So let us start about signal processing. I mean, this term signal processing you have heard so many times. EEG measurement. I mean, there is a cycle actually. You see here, data from brain is signal acquisition. Look at here carefully. It goes to this block called as decoding algorithm, where we decode the data, we interpret the data, and then where does it go? It goes to sending commands, and then this command can be used for different applications. It can be used for computer-aided analysis. It can be used for this wheelchair and other robotic arm applications for industries, but this can also be used as a feedback signal where EEG measurement can be done, rehabilitation tasks can be done. So in the entire cycle, let me tell you one thing which is very common is signal is everywhere. And from one stage to next stage, what is done over the signal? Some processing. So what do you understand by processing? Whenever you talk about processing, means certain operation is performed. That operation may be Fourier transform, that operation may be discrete Fourier transform, that may be some filtering operation, that may be some wavelet transform, that may be any operation, mathematical operation that is performed over the signal. So this is where the signal processing comes into picture. When you capture raw data, you extract features from the raw data. How do you extract features? You use some signal processing method. And then when you pre-process the data, how do you pre-process the data? Again, you use the signal processing. When you amplify the signal, how do you amplify the signal? Again, you use some signal processing as an amplifier. I mean, I'm using a new term here, amplifier. I mean, bioamplifier. Sometimes the amplifier which is used for brain-computer interface, we call this as bioamplifier. Can anybody answer this? I mean, how different it is and why amplifier is needed? Anybody? Please try to answer what do you mean by bioamplifier and why bioamplifier is needed. Come on. Any bioamplifier, any idea about bioamplifier and why it is needed? See what happens that whenever you perform some signal processing task, when you capture a signal, when you record a signal and it goes inside your system, it weakens. The strength of signal is weakened. Even if you convert your natural signal, that is analog signal or what you call as a continuous signal, when it is converted into discrete signal, so what do? You sample the signal, you quantize the signal, you encode the signal. So during these operations, the strength of the signal goes down. So therefore, weaker signals you cannot send to the classifier. You need strong signals, and therefore you need to use amplifier. So be it pre-processing, be it filtering, be it acquiring, be it removing artifact, be it classifying, be it amplifying, everywhere there is a role of signal processing. So if you want to say what are the major stages in brain-computer interface, one is the signal processing, second is the artificial intelligence. Of course, signal processing and artificial intelligence are the major blocks which are used in brain computer interface. I hope I have already explained about this thing, but I think this picture is coming first time. Look at the PET. Where is the temporal resolution highest? Let us take to all the points. Where is the temporal resolution highest? In the PET, because this records your metabolic chemical processes. Where is, I mean, what about the spatial resolution? Spatial resolution is here. I mean, this is the spatial resolution. This is really functional MRI has a lesser temporal resolution than PET, and then microarray has the temporal resolution. And please understand, if this value is less means what? If this value is less than means what? It is in terms of millisecond. So microarray means it is inside your brain. So the total time taken is very less. That is the meaning. And when you are recording PET, it is taking so much of time. 10 to the power 5 milliseconds means it is 100 seconds. almost 100 second it is taking. When you say almost 10 to the power 2 millisecond means 0.1 second is required to capture EEG, MEG or ECOG, almost same. And this is a comparison between spatial resolution, that what is the distance between the 2 points, two adjacent point. So it is decreasing when you are using microarray. When you are using ECOG, I have already explained, that is, it is a semi-invasive, and it is highest in case of EEG. So various chemicals can be imaged with PET. I have explained about PET. Temporal and spatial details are very poor when compared to images produced by this one here. Imaging of electrical processes can give excellent temporal electrical details. It's not excellent, but medium value. I think this is not a new slide. This was already appearing in the previous one. You see the EEG comparison in terms of spatial resolution and temporal resolution. Look at here. Spatial resolution is very high in invasive. High means value is not high, value is less, the distance is very less. Remember, if you go to the diagram, very high means value less. So here it is very high. Temporal resolution is also high. So you can have a comparison. ECOG still spatial resolution is high. EEG it is low, but still, the signal level is not very weak, that you can use for mathematical modeling, and you can interpret the brain signal. That is what we do. So this is another modality called as, sometime we use another term called as functional transcranial Doppler. It measures the changes in the blood flow. We have used the terminology called as blood flow in the previous slide. So how do we measure the blood flow? With the help of FTCD, and in the major arteries of brain, you can measure the blood flow with the help of FTCD. Isn't it? The equipment is mobile, portable. You see here, there is a probe which you are wearing, which a person is wearing, and it is affordable. It can be performed with a small probe on side of the head. This is quite sensitive and can only measure the difference between left and right hemispheres, not useful for BCI purpose. So it is not very useful for the BCI purpose. It has a limited application. I have already, explained about the difference in brain signal. So in a previous diagram, there was a picture. Look at the picture. When a person is having Parkinson's disease, there is, blood flow is very poor. But here in this case, you look at the blood flow in the normal brain. So this is what I was trying to tell you. How does BCI help in classify? You take a lot of normal people, you take a lot of abnormal people, and find out the difference in terms of blood circulation, blood flow, with the help of different images. And this is how you can classify that 30% people, their blood flow is very less. Now in a group of 30 people, again you can classify, in which case blood flow is lowest, in which case blood flow is lower. And again you can correlate the blood flow with the age, with the gender, whether girls or boys, or whether young or old or adult or a child. You can accordingly study about the blood flow with the help of PET scan. So PET scan tracks blood flow. I have already explained, with the help of metabolic process. It is slow. Remember, see the diagram. I'll take you to the diagram again. PET, the temporal resolution and spatial resolution. Imagine in PET, it is 10 to the power 5. So almost 100 second is taken. And therefore it writes, look at what is written here. It's very slow. Time taken is so much. It requires injection of radioactive compounds. Do you remember what is the name of radioactive compound? In the previous slide, it was mentioned, marker. Marker is something that you insert, inject, and with the help of marker you record the what is called as PET. Less useful for BCI application, because in BCI application speed is important factor, and speed is less in PET, and therefore it is not very popularly used. Speed and ease of use are very important factors when we want to use any brain signal for BCI. So PET is not very popular, isn't it? There is one more called as functional near infrared spectroscopy. You understand the meaning of spectroscopy? Spectroscopy is something that measures some light intensity. So therefore, in a picture, look at there is a light source and there is a light detector. You know, the example of light source is LED. The example of light detector is photo detector. Hope you know that laser and light. I'm sorry, laser and LED are the two big examples of light sources. Light sources are the sources that can generate the energy in the form of light. And where what do, what are the names of light detectors? Photo detector, photodiode, phototransistor. These are the examples of light detectors that can capture the light which is coming from other part. So the principle of working of this modality is it measures the blood flow. This also measures the blood flow, remember, by tracking the changes in the different forms of hemoglobin. How does it track the blood flow? With the help of hemoglobin. How? It changes in the hemoglobin accompanying brain activities. I mean, whenever brain activities are there, you know that brain activities are involving oxygen flow and the glucose level. So whenever the blood flow is there, hemoglobin level will be there. So the change in hemoglobin, that is called as blood oxygen level dependent bold response. So when you detect that blood flow level, and the response is called as bold response. What is bold response? It is nothing but blood oxygen level dependent response. So this modality uses infrared light of specific wavelengths that passes through skull. Look at here. It can pass through skull, reflected out where it is detected. Again, light goes inside and comes back, and you record the time and all that. Relatively easy and convenient to use. And again, it can be used in BCI again, but it's not very popularly used in the BCI. There is another term called as functional MRI. You might be hearing this term again and again from me. That functional MRI is also very popularly used for other purposes. This is very expensive, remember, very expensive. If you, I think, in my opinion, if you want to, get your functional MRI from a clinic, from a, radiology center, I don't think it will be less than, $100, for one functional MRI. It will be more than €100 or $100, isn't it? So it's expensive. Functional magnetic resonance imaging. What is MRI? Magnetic resonance imaging. MRI is also used for capturing different parts of body, different organs of the body. So functional MRI, it measures the bold response. What is bold response is already explained in the previous slide. It is blood oxygen level dependent response. It's called as bold response. So bold response is also measured by functional MRI by tracking the changes in the blood flow indicated by relative amounts of different forms of hemoglobin. Remember, let us not go into much technical, otherwise you will get bored. What is hemoglobin? And there are different forms of hemoglobin. By the way, those who are from that kind of background, like pharmacy and other background, they will be very happy when you are coming across these terminologies like hemoglobin, blood flow, and all that, isn't it? So there are different forms of hemoglobin, and what are the different amounts of those different forms of hemoglobin? Accordingly, there will be change in the blood flow level that is recorded by functional MRI, and this is how the entire functional MRI work. It has been explained here. I think the description has been included. It is expensive, technically demanding. There is technical expertise required to capture your functional MRI. You cannot capture your own, what is called as, the functional MRI. Most sensitive and having highest spatial resolution. Remember, functional MRI has highest spatial resolution, other than that, the chip. So how do we measure the blood flow response? The oxygenated blood is pumped from the heart. How do you get blood into the brain? It is pumped from the heart through arteries to the smallest blood vessels and capillaries that pass through all the organs and muscles. That is the fundamental level. It releases oxygen supporting metabolic process and absorbs carbon dioxide produced by metabolic process. So it also absorbs carbon dioxide and releases the oxygen. How does it absorb carbon dioxide? Because carbon dioxide is produced by metabolic processes, otherwise it will damage. So the blood returns to the heart via veins and the circulates through the lungs where it is releases carbon dioxide and picks the molecular level of oxygen. So let us not go into the detail. And then that blood returns, oxygenated blood, it goes to heart and comes, returns to the heart, and cycle starts again. Each cycle takes about 1 minute. In the blood, transport of oxygen, this is important. In a blood, transport of oxygen is carried out by, you all know, hemoglobin, an iron containing complex protein that can bind the oxygen. So that there are different types of hemoglobin. It exists in the deoxyhemoglobin form, which is written as deoxy-HB, which does not contain bound oxygen. I mean, the oxygen which are binded together. And oxyhemoglobin that does have bound oxygen. So this is the difference. So what is the name? What is the type of hemoglobin? What is the amount of hemoglobin? That is also tracked in the blood flow by using the technique called as functional MRI, magnetic resonance imaging, and that too functional. So as the blood passes through the lungs, each deoxy hemoglobin molecule picks up four oxygen, molecules and thereby becomes oxyhemoglobin. When the blood passes through organs and muscles, oxyhemoglobin releases the oxygen and becomes deoxyhemoglobin. This is a phenomenon. When the brain region, now coming to brain, when a brain region metabolic activity increases, as we have mentioned in the PET also, whenever metabolic activity increases, it oxygen level, consumption increases. It requires more amount of energy. About a second, the neuron begin to absorb more oxygen from oxyhemoglobin in the blood of nearby capillaries. So it will ask for more oxyhemoglobin. When brain activity returns back, suppose your brain activity is coming lower to the baseline, the three properties also revert to back of the origin. All these properties will also revert back. So that tracking happens. And during the process, what is important when, something is going high and becoming low, you generate some signal equivalent to this one. So this is also true in case of EEG called as, action potential. Changes in the communication between neuron. When neuron is communicating between their groups, that is recorded in terms of firing rate or local field potential. Potential is nothing but electrical voltage with the microarrays or measuring large scale potentials with the help of EEG. So this is how, it fires whenever firing happens. Suppose, group of neurons interact and they fire each other. The moment they fire, there will be some peak, and this particular peak, will be recorded. And I mean, there will be some negative voltage, there will be some positive voltage. All kinds of voltages, variations are recorded. Small variations are recorded, and that variation is known as either your EEG signal or ECOG signal. And the basis of working of brain computer interface, complete working, BCI, that is based on this phenomenon. You generate active potential, it is recorded in time series in a continuous manner, you get EEG or any other kind of signal. So that reports communication and interaction, the brain activities of those neurons, which is recorded. So changes in communication can also be measured using either functional NIRS or functional MRI. Because whenever they communicate, those neurons, say, when you are trying to solve some critical, complex problem, what will happen? Your brain activity increases. Whenever brain activity increases, there might be more and more interaction between groups of neurons. And therefore, the functional MRI can also track the changes in the blood flow in terms of the types of hemoglobin, the amount of hemoglobin, whereas the PET can also measure the blood flow tracking. So in all the cases, the changes are recorded in terms of electrical potential. Now, let us understand, because we are talking about EEG signal. So let's understand, the signal processing little more using, EEG signal. So you look at one picture. This is a montage. Generally, we call it a montage. Montage means configuration of electrodes, configuration or structure of electrodes. Look at here, NZ, IZ. This is A1 and A2. Can you guess where are the ears? ears, that is human ears. Where are the human ears? Can you guess? I mean, which are the
One,
nodes?
two.
Oh, very good. Have you worked with EEG before?
Nope.
So how do you get an idea?
Because there is a triangle near the
Yeah,
NZ,
yeah. Thank you.
and
Very
that's the
smart.
nose.
Yeah, yeah.
And
Thank you.
so the one that circles under the
Thank you.
A1 and T.
Thank you. Very smart answer. Of course, N is also indicating for nose, nasal side, and these two are ears, and the symbol of ears are also written. So generally, A1 and A2 are the electrodes. They are connected to ears as the reference potential. Yesterday, I was telling you about the reference potential because when you want to measure the potential of F10, for example, so a channel consists of two wires. One wire will be connected to F10, and the difference between the difference between the potential of F10 and A2 will be recorded as the potential difference of F10, and therefore you always need the reference electrodes. So here your A1, A2 are reference electrodes, and all these are channels. And the concept is same here. I think let us not repeat. Central point all Z, this side all even numbers, and this side all odd numbers. And I have already explained about delta wave, theta wave, and alpha wave. When you compare delta, theta, and alpha, you will find that alpha is having highest frequency. If you compare between the three, theta has higher than delta, delta has lowest. And let us understand the properties of signal. This diagram, can you imagine, is representing VEP, is representing VEP, or sometimes we call it as a ERP rather than VEP. Let us call ERP. What is ERP? Is event related potential. Event related potential. Is it different from EEG? It's a kind of EEG. But what is event related potential? Event related potential is specific to an event. Suppose you are watching 0 for 10 minutes, you are watching 1 for 30 seconds. So when you are watching 1 for 30 seconds, your brain responds and generates certain signal. And that is what is called as event related potential. Now, what is this N1, N2, P1, P2? We'll explain, but obviously you can understand very clearly. P1 is a positive peak, first peak here, positive peak. P2 is a second positive peak, this side positive voltage. Look at the diagram carefully. The upper side is negative, the lower side is positive. You can reverse the diagram also, there is nothing wrong. So this side is P1, P2, and this side you look at here, N1 and N2, which are negative. There is one more term called as NPAT. There will be N1, N100, P300, P200, N200. What are those P300, P100, P200? Let me explain. And they are very, very important for BCI applications, especially P300 BCI, isn't it? Now, this is a normal signal processing where signal is recorded, amplified, and then you get different samples. Those samples are recorded. So each sample here is of 1 second, and all these are connected, and you get an EEG signal. So what do we mean by properties? It can monitor the human brain activity non-invasive manner with the precision of millisecond. Even with the precision of millisecond, we can monitor. The recordings can consist of oscillations. These are nothing but oscillations because it does not generate a single potential. In a continuous manner, it generates the potential. So therefore, oscillations, the moment you call frequency, frequency means oscillations are there at different frequencies fluctuating over time. Look at it is fluctuating over time and provide meaningful information about brain states. What is the state of brain? What are the activities where brain is engaged are all recorded, isn't it? Brain oscillations such as alpha frequency, alpha, beta, theta, all are responded. Yesterday I have explained you 1 to 4, 4 to 8, 8 to 13, 13 to 34, more than 34. 0 to 4 is your what is called as theta, delta, and then 4 to 8 is your the theta, and more than that is alpha, and then comes beta, more than 13, and probably up to 30 is beta, and the more than 30 is your gamma. So there are different ranges of frequency. Event related potential does not necessarily capture all the event related information present in the EEG. Rather, it is very specific to certain event. To extract more information related to sensory, cognitive, and motory, ERPs are used. What do you mean by sensory, cognitive, and motory? Any idea? Sensory means related to your ears, eyes. You are sensing something, very specific event you are realizing and you are capturing. Cognitive means you are either learning some skill, you are developing, you are improving some skill. And motory means you are moving your hands, your legs, your different body organs, you are blinking your eyes. So that is called as motory. So there are sensory events, there are cognitive events, and there are motory events. Each event will cause the electrical potential to be generated by the brain from a particular part using a channel, using a sensor, is called as is ERP, called as event related potential. So let us further explain about ERP. ERP components are very small in magnitude. Their range is 1 to 20 millivolt, and inter-individual variation is susceptible to various artifacts. ERP components are represented by letter N. I have told you N and P are used. Look at here, P1, this side N1 is the first N1, P2 is P2, N2 is second N2, where 1 to 3 are actually 100, 200, 300, 400 millisecond. So when you say P1, it is also written as P100. When you say N1, it is N100. Same thing. When you say P2, it is actually P200. When you say N2, it is actually called as N200. And P3 is actually your P300. So what is N? It is negative polarity. What is P? Positive polarity followed by a number. And what does that number represent? That is very interesting. Is the latency. What is the latency? You see here 100 millisecond means you will start capturing the event. Suppose you are watching a candle is burning and you are watching it continuously. The flame you are watching continuously. So, how much time it takes to produce first peak, positive peak? It is increasing. Now you have reached to peak point. How much time is the latency, is the delay? So, latency is it follows the number. So, when you say N1, 100 millisecond is the latency time. So, P1, 100 millisecond is the latency or the component's ordinal position in the waveform. So, for example, say the substantial negative peak in the waveform occurs 100 millisecond after a stimulus is referred as N100. After a stimulus, remember, when you want to focus some object and the object is not appearing, the moment it starts appearing, from that time your stimulus starts. So whenever stimulus starts appearing, from there, how much time it takes to reach to the peak value, either positive side or negative side, accordingly you use the terminology P100, P200, P300, and so on. So how does it work? You have a stimulus or a stimuli that is sensed by sensory organ. The stimuli may be very less in strength, that may be weak. So what do? You amplify, and then you use some other signal processing. You can perform averaging, you can perform mean value and all that, and then you can find out the peak value. So this is how ERP is used. This is not that ERP computer science people study. Those two who are masters in computer science engineering, do you know some other name for ERP? Yeah, I think Maria is from Master in Computer Engineering. I mean, is Maria present? Would you like to answer this? In computer science and engineering, there is one more term called as ERP. ERP stands for what? Anybody can answer without using your mobile phone. ERP in computer science and engineering is what is ERP? Any idea about ERP? Come on, someone, I'm just waiting. I do not want to, do one-sided, non-stop. You'll get bored. And by the way, today is the last lecture of online, so do not get bored. I hope you are uh, you may not be enjoying much, but you may be getting exposed to new area. If you have not studied before, you must be getting exposed to new area, new knowledge about, wow, this is also an interesting area, this is a very interesting area connected to our brain. I mean, I hope that it is creating an interest in all of you. Yeah, I asked some question. What is ERP? You do not want to answer? Someone, I mean, two people have already left, thinking that now professor will be closing soon. When we started, there were 9 people. Now we have only 7 people. Anybody wants to answer? What is ERP? I'm just asking to give you pause, you know. I think I mentioned the name Maria. Maria is not present today. Present. Uh, anyway, okay. If you have decided not to respond, it's okay. I think
I, I know it's wrong, but enterprise resource planning, it's what comes into my mind. It's totally
so
not,
why you are
not
no why you are saying, who is answering, by the way? What's your name? Because I'm in the slideshow mode. Who is answering this question?
Federica.
Yeah, Frederica. Why you are saying answer may be wrong? Your answer may be absolutely correct. In computer science and engineering, ERP is enterprise resource planning. All organizations, institutions, they have their digital, organization system, digital campus system, digital, way of working. And without ERP, an organization and industry will fail. So that is ERP, enterprise resource planning. You are absolutely correct. Why you are saying it may be wrong? Yeah, it's not wrong. It's true. Okay. So the idea is related to brain computer interface. It is not enterprise resource planning, rather it is event related potential. Why it is called as event related potential? Because the signal, that is voltage signal, is related to a specific event, and therefore it is called as event related potential. Now I was using the term P300 BCI. This is used for, this, especially the 300 component, is a component between 250 millisecond and 700 millisecond. This 300 component is very, very popular. Now one can ask this question, professor, sorry, I just want to go to previous slide, I'm sorry. You can say that, professor, why we are only capturing 300? Why not 250? Why only 300 BCI? P300, why not P200 and P100, BCI? The answer is very simple. Here the strength is highest. P300, the strength is highest. So better to, consider the higher value, highest value, so that it can convert the meaningful interpretation of signal. It can convert the signal into commands. It can translate into command and very easily. Therefore, the highest one which is considered, and generally it is between P300, occurs between 250 millisecond and 750 millisecond. That is written there. You look at here, anywhere between 250 and 700. ERPs are caused by processes like memory, where exactly ERP, you are trying to recall something, you are trying to pay attention, or changes in the mental state. The processing of physical stimulus is reflected by evoked potential. Physical stimuli means anything which you are concentrating, anything which you are sensing is a physical stimuli. That may be a light, that may be a darkness, that may be a sound, that may be a clicking sound, that may be a flickering light, anything which is, causing you a stimuli in front of you. All right. ERPs are used in neuroscience, cognitive psychology. This is a general application, not the P300. ERPs are used in neuroscience, cognitive psychology, psychological research, dementia, Parkinson's disease, stroke, head injuries, obsessive compulsion disorder, OCD. They have some abnormalities, and those abnormalities can be reported by ERP components. ERP components can be used to study dementia, Parkinson's disease. Why? Can you guess? The answer is, see, in dementia, it is related with memory. Somebody forgets, not all the time forget, sometime, and therefore it is related with event. A person doesn't recall something, the person doesn't recall some event suddenly, so it's related with event, it is related with memory. So It is used to study, it is used to diagnose these diseases like dementia. This is not disease actually. With a dementia, person is not dying. The person is still surviving, person is living. It's a disorder or disability. Dementia, Parkinson, stroke. I mean, if these are very severe brain stroke, very severe head injuries, then a person will die. Otherwise, with a little less stroke, with a little less head injury, person will not die, but might lose some brain's ability. That is true. So all these studies, research are performed with the help of ERP, rather can be performed using ERP. It's not only ERP, but you can use any other EEG signal or functional MRI, another signal to study about stroke and head injuries also. But by the way, your ERP is nothing but an example of EEG signal only, subjected to a particular event. So in the recent past, P300 is used to develop BCIs in which the subject can communicate with the stimulus he is looking at. What I'll do that, because today I have not included the a practical application of B300. So what I'll do that P300 BCI. Let me try to get. I think there was some paper. I think I have a YouTube also. Very good. And is this? Let us see that thresholding based. Hope this. Wow, very good. This is our own paper. This was published in IEEE. When it was published in year 2021. And the name of journal is, it's not bad journal, IEEE censor journal. It is not transaction. It is not, conference proceeding. It was a journal. And look at the title, thresholding free state detection approach for home appliances control. The person who is having certain disability can control different home appliances using the P300 based BCI. You can read about this paper later. I think this is a picture. Can I increase this? This is a abstract where you can understand how does it work. So this is so interesting. B300 is very very interesting area of the B300 BCI. Now, I was talking about different types of analysis of EEG signal, time domain analysis, frequency domain analysis. So, time domain analysis means directly reading the EEG signal and doing analysis. So, we generally do not prefer time domain analysis. We always use frequency domain analysis in any signal processing because it's a spectral analysis. It gives frequency versus amplitude. At what frequency, what is the amplitude of signal? Look at here. This side is a frequency, on x-axis, and y-axis is a normalized amplitude. This may be either energy or power or amplitude. So that tells the variation of a signal over the frequency range. And therefore, frequency domain analysis is very popularly used. You can use any type of Fourier transform, any type of transform that converts time domain signal into frequency domain can be used for, doing the frequency domain analysis of, EEG signal. So when you get frequency domain, when you convert your EEG signal into frequency domain, you can also calculate PSD. What is PSD? Power spectral density. Now, you read any research paper using EEG signal. In most of the papers, the common attribute of brain signal will be PSD, I can tell you. Most commonly used attribute that is used in all the EEG research papers is power spectral density. What does it do? It reflects the frequency content of a signal or a distribution of signal power over the frequency. So this is the amplitude spectrum. But what is PSD? It is a power versus frequency. Or at a particular frequency value, what is the power level? What is the power distribution of a signal? That is indicated by the PSD. It's very useful. Time domain analysis is directly watching the EEG signal and using some linear predictive component analysis and analyzing. And that is not correct way. It is unsupervised method. I mean, where is the end? You don't know. Where is the destination? You don't know. So it's not very popular. What is popular is the what is called as frequency domain. And what is the name of time frequency analysis? We use the Veblet transform that captures that gives us spatial as well as temporal results. And then we can use ANN, and we can classify the signal in a proper way. These are the attributes: peak to peak, mean square value, variance value, maximum power spectral density. All these linear and non-linear parameters can be used to train the model. I've already explained you what happens that when you acquire the brain signal, you pre-process the brain signal, you have to extract some attributes. So these are the attributes, because your ANN model, your deep learning model, requires some attributes. And from where you will get attributes? Using some wavelet transform, Fourier transform, or any signal processing model, you will get these parameters, attributes. Why do you need attributes? Because attributes will be the input for the machine learning model. Machine model, ML model or AI model do not understand pictures, do not understand EEG signal. They understand attributes. So you need attributes. That's all. But yeah, CNN based model, deep learning based models, you can directly give the signal as an input. They have a capability. They will extract the features. They will classify them. All these are inherent in the deep learning model. I believe that all of you are aware about deep learning model. Deep learning model has a capability of pre-processing. You do not need to even use the pre-processing method separately. You can still use pre-processing if you're using deep learning model. But even if you're not using, CNN or deep learning based model takes care of pre-processing, signal processing, removing some noise, extraction of features. All these are taken care of. And then we do some signal to noise ratio analysis and all that. So let us not make hurry. What I'll do that we'll stop at this particular time, so that we can start from the next lecture from what are the other parameters which are used, and then with the help of practical examples we'll understand how the signals are recorded, how the signals are interpreted, how signals are utilized for different applications, be it mindfulness practice, be it P300 BCI, or be it some other applications of BCI. So I think we have another how much, 3 minutes. So if you have any question, because today is the last day for meeting online. So if you have any question online, you can ask me online. Otherwise, we'll be available offline soon. Yeah, anybody? Oh, you are very excited to say bye bye. My 2 hours was a very lengthy hours. Oh my God, so much time professor is speaking. Let us say bye bye. Is it true?
No, not
Is it
really.
true? Is it true? I mean, you are only speaking. What about others? I mean, anybody? Is it true that the lecture was very boring and you are waiting for closing the lecture? Not like that? Okay. All right. I mean, you do not prefer responding. All right. So thank you so much. It was very nice to meet you online, and take care of yourself. And looking forward to see you in your university. Hope you too, look forward to welcome me in your campus.
Yes, yes. We are very
Others are
excited.
others are not saying anything. They're not excited to welcome me. They have decided not to speak, even not to speak about saying you're welcome, professor. Anyway, I know that, yeah, you all are. I'm very happy that you attended all these three online lectures. Maybe by this weekend, I will send you some task, very small and simple task for your homework and assignment. You will start doing that assignment. When I come there, I will discuss those assignment with you, and we'll have a PowerPoint presentation in your campus, and will give you grading also, so that you can get 4 CFU after attending this particular course. Thank you so much. Take care. Bye bye. Bye.
Thank you. Thank you. Bye bye.
Thank you. Bye.